Testing, 1, 2, 3. Excellent. All right, we're going to get started in about a minute. Appreciate everybody being here, and I know things work on Adventist time around here, and most places. Even the clock when we came in was on Adventist time. We noticed by the ABC, it was like 5 minutes late. How they even tell their clocks to be on Adventist time around here. I always start my classes right on time, though, so that way if they come late, my students miss out on a little bit. All right. All right, well, it's good to have you guys here and be back one last time. My coat got dirty this morning because of doing a lot of demos. I'm doing a lot of cool demos with the junior tent. I should bring them over here. They're in my bin over there. All right, I see that it's 11 o'clock, so it's probably time to get started. And as always, I'd like to start with a word of prayer, and we'll get going. So let's pray. Father God in heaven, we're so thankful for another day of life, for what you have blessed us with in this world, this amazing place that we're able to live, to breathe, to grow, to have freedom, and to enjoy what you have made. But Lord, this world is not without, it's not just all enjoyment, and we know that. There's a lot of suffering that takes place. And you're the true antidote to the suffering that's around, and we thank you for that. And as we explore your world in a little bit more detail, please bless the words and our thoughts here today. And we do all this for your glory and your honor. And we ask the blessing now in the name of Jesus. Amen. All right, today I'm going to talk about the top 3, I think, arguments or scientific problems that the theory of evolution really has. I don't see, and I've been looking, I don't see any really good arguments. They say they have awesome arguments to defend these points, and we'll leave it up to you all to see if that is true or not. And so I call it well, because there's 3 things. So I'll start it with a joke. It's not necessarily a great one, but you can decide that too. Did you hear about the 3 holes in the ground? Well. Okay, you guys are very nice. Not a great joke, but so I want the idea of wells, a hole in the ground, to be in your mind for a few moments. Because the problem with being in a hole and with a hole is if you get stuck in the hole, how do you get out? And if you don't get out, eventually you die. And so if your argument and your idea is stuck in a hole, I think then your idea may die. So 3 main areas I want to talk about today is the fine-tuning of our universe. I've been talking about the fine-tuning of our world, and I want to show you, actually each one of these we have a little video to go along with them, and then I'll just do some discussion along with that. So the fine-tuning of the universe, that's one major argument. Second, how did life get started? What is the chemical origins of life? And then lastly, we're going to talk about information and words and ideas and codes. And I think these are probably the 3 biggest things. And what I've been doing all week is probably part of this first idea. So the fine-tuning of the universe, chemical origins of life, and information, especially that which we find in our DNA and RNA. And we're going to discuss these 3 sections here over the course of this morning. And so to get things started right off the bat, some of these things people have done already, and they've done a great job, and then I'll build upon it. And so there is a organization called Reasonable Faith, and they have produced a video on the fine-tuning of the universe. It gets into the physics and the mathematics, which I love. It's awesome. They have done a great job with the animation, and I'm really just going to turn it over to them. I think the video is 5 or 6 minutes long, and we're going to let the Reasonable Faith and their video on the fine-tuning universe get us started this morning. So I'm trusting that the video is ready to go.
From galaxies and stars, down to atoms and subatomic particles, the very structure of our universe is determined by these numbers. These are the fundamental constants and quantities of the universe. Scientists have come to the shocking realization that each of these numbers has been carefully dialed to an astonishingly precise value, a value that falls within an exceedingly narrow, life-permitting range. If any one of these numbers were altered by even a hair's breadth, no physical, interactive life of any kind could exist anywhere. There'd be no stars, no life, no planets, no chemistry. Consider gravity, for example. The force of gravity is determined by the gravitational constant. If this constant varied by just 1 in 10 to the 60th parts, none of us would exist. To understand how exceedingly narrow this life-permitting range is, imagine a dial divided into 10 to the 60th increments. To get a handle on how many tiny points on the dial this is, compare it to the number of cells in your body, or the number of seconds that have ticked by since time began. If the gravitational constant had been out of tune by just one of these infinitesimally small increments, the universe would either have expanded and thinned out so rapidly that no stars could form and life couldn't exist, or it would have collapsed back on itself with the same result: no stars, no planets, and no life. Or consider the expansion rate of the universe. This is driven by the cosmological constant. A change in its value by a mere 1 part in 10 to the 120th parts would cause the universe to expand too rapidly or too slowly. In either case, the universe would again be life-prohibiting. Or another example of fine-tuning. If the mass and energy of the early universe were not evenly distributed to an incomprehensible precision of 1 part in 10 to the 10 to the 123rd, the universe would be hostile to life of any kind. The fact is, our universe permits physical, interactive life only because these, and many other numbers, have been independently and exquisitely balanced on a razor's edge. Wherever physicists look, they see examples of fine-tuning. The remarkable fact is that the values of these numbers seem to have been very finely adjusted to make possible the development of life. If anyone claims not to be surprised by the special features that the universe has, he's hiding his head in the sand. These special features are surprising and unlikely. What is the best explanation for this astounding phenomenon? There are 3 live options. The fine-tuning of the universe is due to either physical necessity, chance, or design. Which of these options is the most plausible? According to this alternative, the universe must be life-permitting. The precise values of these constants and quantities could not be otherwise. But is this plausible? Is a life-prohibiting universe impossible? Far from it. It's not only possible, it's far more likely than a life-permitting universe. The constants and quantities are not determined by the laws of nature. There's no reason or evidence to suggest that fine-tuning is necessary. How about chance? Did we just get really, 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 really lucky? No. The probabilities involved are so ridiculously remote as to put the fine-tuning well beyond the reach of chance. So, in an effort to keep this option alive, some have gone beyond empirical science and opted for a more speculative approach, known as the multiverse. They imagine a universe generator that cranks out such a vast number of universes that, odds are, life-permitting universes will eventually pop out. However, there's no scientific evidence for the existence of this multiverse. It cannot be detected, observed, measured, or proved. And the universe generator itself would require an enormous amount of fine-tuning. Furthermore, small patches of order are far more probable than big ones. So the most probable observable universe would be a small one, inhabited by a single, simple observer. But what we actually observe is the very thing that we should least expect: a vast, spectacularly complex, highly ordered universe, inhabited by billions of other observers. So even if the multiverse existed, which is a moot point, it wouldn't do anything to explain the fine-tuning. Given the implausibility of physical necessity or chance, the best explanation for why the universe is fine-tuned for life may very well be it was designed that way. A common-sense interpretation of the facts suggests that a super-intellect monkeyed with physics, and that there are no blind forces worth speaking about in nature. The numbers one calculates from the facts seem to me so overwhelming as to put this conclusion almost beyond question. There is for me powerful evidence that there is something going on behind it all. It seems as though somebody has fine-tuned nature's numbers to make the universe. The impression of design is overwhelming. The heavens declare the glory of God. The skies proclaim the work of his hands. Day after day they pour forth speech. Night after night they reveal knowledge.
It's amazing to me. And I love that because it gets into the mathematics, the physics of the universe, and you can choose to, agree with this video, or you can choose the other options that everything is perfect and perfectly works out for life. Because, I think there's a lot more scientists that are buying into the idea of the multiverse. And that there just had to be, if there's so many universes out there, one of them had to work out. And I see the scientific community sort of picking that up as the major defense against this awesome fine-tuning that we see. Well, of course, it had to be. Eventually, one of the universes had to create. So I really appreciate this organization, Reasonable Faith, not an Adventist organization, but there's a lot of intelligent design, creation organizations. We're all kind of working in similar directions. A number of the intelligent design communities, they're not always Christians, they may be atheists, but they see scientifically that design is one of the best arguments. And what I've been trying to do this week is show that the earth, when we look at all the different components of earth, we see all of the same fine-tuning with the air and all the ingredients in the air. And we look at the magnetic field and lack of electric field, Earth's gravity, and some of the chemistry that takes place. We see the things that are there and the things that are not here as giving life. Now, we're not without our problems, but the problems on a large scale are somewhat minor in the sense that, yeah, we do have problems of hurricanes, tornadoes, and we do have these things, but we actually have a lot of knowledge to be able to protect ourselves from them. So they may seem like major issues, but when you're trying to create a universe, try to create a planet that's hospitable to life, this becomes a big challenge. And so it's amazing to look at even our position in not only looking at our Earth, but looking at our position in the solar system, our solar system's position within the Milky Way. I'm not going to get into a lot of those details, but this fine-tuning is amazing. And maybe even our Milky Way's location within the grand scheme of things within a supercluster that they're naming Laniakea that we're a part of. It looks like even that location of the Milky Way and the supercluster we're a part of seems to be even finely tuned. It just keeps going. We're in a nice, safe neighborhood to allow life to exist. So how do you get out of this hole? Well, it feels like some of the explanations, like multiverse, this is an analogy I heard from actually Stephen Meyer, who works at the Discovery Institute here in Seattle. And he likened it to, if someone's stuck, you could just go, if you're stuck in a hole, just go to the garage, get yourself a ladder, and climb out with the ladder. But how did you get out of the hole in the first place? And so there's some circular arguments that seem to be there. And the other thing I want to impress upon you, that it's not just like, there's no other explanation that it has to be a designer. And as we get through this discussion today, I hope to change it from, there's no other really good explanation, we just haven't done enough research, to, oh no, this is actually the exact way that we see intelligence and design working. All right, so that's the first argument, and it continues that there's a lot of debate going on in between that. I just wanted to give you that first thing. Now, as we get into the second topic of, chemical evolution, how did life get started on this planet? So we could say, all right, we'll give you the argument number one for those that believe in a naturalistic view of life. Okay, I'll give you that somehow we're in there. Well, how did life get started? Do we have any good explanations of how did the chemicals get here on this planet, and how did they come together and form life? And so as the scientists have been looking in these various fields, physics for a long time has said, I mean, look at the light from these stars. We know that the stars are billions of light years away, and so the universe is it's very large, 14 billion light years across, something like that, and that we have this huge universe. And so physics says, oh, look, we've got the time to build complicated creatures. And biology says, look at all these creatures, and we see them changing, adapting, and if we can just dial it back, well, they must dial it all back to a simple creature, a common ancestor. And it's really been the missing link in all of this, actually, I think, is chemistry. Because in order to get life started from the elements, you have to think about the chemistry. And chemists have not been getting into this discussion because we've been busy doing other things. People want the world, chemists, hey, can you analyze our water? Can you analyze the air? Can you analyze the food? Can you go build lightweight this, less toxic this? And so chemists, we're busy making, analyzing, dealing things with energy, and so we don't have a class in our department that says, well, what's the evolution of chemicals that has taken place? We don't have that. In most biology departments, you have a class on origins or theories of evolution. They will discuss it in one way or another, but that's usually like a big thing. We don't discuss that in the chemistry department. Now, some may be starting to discuss it more, as I will describe. I don't have that class. Nobody teaches that class in the chemistry department. It's not been an important thing. We're still trying to figure out how molecules still interact. We have some great theories, and we're pretty good at making things, but in general, we're still inefficient on how to bring chemistry together. So the question then becomes, well, is chemistry then going to show us the doorway of how all these chemicals came together to make life? Or is chemistry actually showing us a barrier that's there? And so then how would you get over that barrier? So this is why you don't hear a lot of chemists get up and talking about these things. This is changing. In fact, our schools, universities, the governments, that's the agencies that are funding science, know that this is a big hole, that this is a big gap that needs to be filled. And so they're creating the Center of Chemical Evolution, throwing in millions of dollars, maybe somewhere between $5 and $20 million a year, I think, to fund universities to do the origins of life, chemical research, how do chemicals come together to make life. And so this is really a recent thing in the last maybe 5 to 10 years. Now, as I'm going to show you, scientists have been thinking about this for a long time. And we'll see some experiments, and one in particular, the Miller-Urey experiment, where they think they've made some important life components, and we're going to talk about that. But why would you need to fund a bunch of research to figure this out if it wasn't a problem? And that's one of the points I want to make. This is a problem. Scientists have no good idea how chemicals come together to make life, because there's some really major hurdles there, some very major challenges. And so chemistry now and how chemicals come together to form parts that are in living systems, this is where a lot of research is going to come. So there's going to be a lot more articles that come out, and it seems like most of them are entitled, "We've solved the chemical origins of life." But you can't have every article having the same title that says we've solved it, because why would you then? You need to have another article by the same title. And so that's often the press releases that come along with there. So chemistry then is, I think, a battleground, and that's why it's important for a lot of us that are in this field to understand the arguments that are there, not to just blindly accept them. Let's use our chemical knowledge to see what's there. Now, back in the 50s, there was an experiment done by a graduate student and faculty member, where they thought about what would it be like if there was an early Earth, what would the air be like in the water and everything and the chemicals? Could we fashion a way to create life or create some of the chemicals that are important for life? And they were able to do this. And this kind of shocked the scientific community, and it ends up in a lot of textbooks. And I think it's important to understand this experiment, because you should hear it. And I think our chemistry and our biology faculty really need to understand this experiment. If it sits in the textbooks, and if you don't understand it, then and how to understand what they were able to do and what it did not do, then we have a problem. So I'm thankful to this website that was funded by National Science Foundation and NASA, and they do this thing called Stated Clearly. And they're going to describe this Miller-Urey experiment. And so our second video here this morning, also, I think it's about 7 minutes long. They do a good job. Great animation. So as soon as I get more funding, I'll get my own videos here. But they do a great job. So we're going to roll the second video on the chemical origins, the Miller-Urey experiment.
Stated Clearly presents... What was the Miller-Urey experiment? It was once believed that if you left food out to rot, living creatures like maggots and even rats would simply poof into existence. The idea was called spontaneous generation. A series of experiments starting in the 1600s disproved this idea, and in the 1800s, a new scientific law was proposed. Life only comes from life. It's true that rats, maggots, and even microbes are far too complex to simply poof into existence, but in 1859, English naturalist Charles Darwin put forth the theory of evolution. In it, he showed that under the right circumstances, relatively simple creatures can gradually give rise to more complex creatures. Given this information, serious thinkers began to wonder, is it possible that simple life forms actually could come from non-living matter? Not by poofing into existence, but through a natural, gradual process similar to what we see in biological evolution. Darwin himself mentioned this idea when writing to a friend. But if, and oh what a big if, he wrote, we could conceive in some warm little pond, with all sorts of ammonia and phosphoric salts, light, heat, electricity, and so on present, that a protein compound was chemically formed, ready to undergo still more complex changes. In 1924, Russian biochemist Alexander Oparin published a book which he titled The Origin of Life. In it, he outlined his thoughts on a gradual progression from simple chemistry to living cells. He imagined the early ocean as a primordial soup, a rich collection of complex molecules produced by natural chemical reactions. In this soup, further reactions could take place, eventually producing living cells. At the time, Darwin's warm little pond and Oparin's primordial soup were really just speculation. They were founded on a good understanding of chemistry and biology, but they could not be considered legitimate scientific hypotheses because no one had found a way to test or observe them. Science, after all, is the study of observable facts and an ongoing conversation about how those facts can be best linked together. Chemical reactions like those proposed by Darwin and Oparin are not expected to leave an observable fossil record. Without either having fossils to examine or a time machine to travel back and observe what happened, how could scientists even begin to study the origin of life? In the 1950s, Stanley Miller, then a graduate student at the University of Chicago, came up with an idea. We could simulate early Earth conditions in the lab and then carefully watch what happens. If you can't study fish in the sea, set up an aquarium. Working with his professor, Harold Urey, Miller designed an apparatus to simulate the ancient water cycle. Together they put in water to model the ancient ocean. It was gently boiled to mimic evaporation. Along with water vapor, for gases of the atmosphere they chose methane, hydrogen, and ammonia. These are simple gases which scientists at the time thought were probably abundant on the ancient Earth. They added a condenser to cool the atmosphere allowing water molecules to form drops and fall back into their ocean like rain. The ancient Earth would have had many sources of energy, sunlight, geothermal heat, and even thunderstorms, so they added sparks to the atmosphere to simulate lightning. The goal of the experiment was not to create life, but to simply test the first step in Oparin's model. Can simple chemistry naturally give rise to the complex molecules of life? After running the experiment for just 1 week, their ocean became brownish black. Careful analysis revealed that through a series of reactions, many complex molecules had been produced. Among these were amino acids, special molecules of life that we once thought could only be built inside the bodies of living creatures. This was a pivotal breakthrough in science, so significant in fact that it gave rise to an entirely new field of research now known as prebiotic chemistry. Scientists don't know for sure if the gases used by Miller really were the most common gases of the ancient Earth. Because of this, many experiments have since been done, showing that the molecules of life can form in a wide variety of environments with different starting chemicals and different sources of energy. Sugars, lipids, and amino acids have even been found on meteorites. This suggests that the molecules of life formed all throughout the ancient solar system and may be forming right now in other regions of our galaxy. Together, these discoveries tell us that Oparin's primordial soup and Darwin's warm little pond could have easily existed in one way or another on our ancient planet. So to sum things up, what was the Miller-Urey experiment? The Miller-Urey experiment was our first attempt at simulating ancient Earth conditions, in this case, the ancient Earth's water cycle, for the purpose of testing ideas about the origin of life. The Miller-Urey experiment is significant for 2 main reasons. First, though it was not a perfect simulation of the early Earth, it clearly demonstrated for the first time that biomolecules can form under ancient Earth-like conditions. Second, the experiment took what was once mere speculation, the idea that life may have emerged from chemistry, and transformed a portion of that speculation into legitimate, testable science. Many questions remain to be answered about the origin of life, but scientists from many nations and many fields of study are now following Stanley Miller's lead. They're finding ways to turn those questions about the origin of life into testable, scientific hypotheses. Simulation experiments cannot tell us exactly how life formed in the past, but if enough of them are done, they could eventually tell us if it's possible for life to emerge from chemistry. I'm John Perry, and that's the Miller-Urey experiment, stated clearly. This video was funded by the Center for Chemical Evolution.
Boy, it'd be interesting to get inside each one of your brains after you hear that. And it looks like, boy, they got it solved in some ways. The experiments were run 1950s, and I don't know. It almost feels like, well, they've got that first part, and I love how they put on their science, bam, and it is. And this test is repeatable. And anybody who can kind of run the same sort of experiment, and you can get amino acids, and there's no problem. That is true. There's nothing wrong with that. So I don't want to get the notion that these aren't good scientists. These are good scientists who are trying to solve a very challenging problem, and it's actually good. I really appreciate that there are scientists who are interested in trying to solve this problem, because it's really, I think we can learn a lot of interesting chemistry. I just think we're going to run into some pretty big dead ends here. And why? What's the problem? Well, I don't know if you caught some of the words to that video. Well, maybe that was not the right atmosphere. Maybe there was something, well, yeah. They said there was a few little things that weren't quite with that. Well, if that sort of solved the problem, why do we keep getting articles that are coming out that say study solves the mystery of the first animals and how they appeared on the earth? And then how did we get, what was this primordial soup like? And there's just a lot of challenges there that I think get glossed over. And we're not going to, I'm trying not to get too deep into it, but we're going to get a little bit into it. Because I think it's important to understand what was wrong and what did we learn. And we're going to bring it back to some of the things that have already been talked about here this week. So they've made amino acids. We can see the analysis that's been done. It's repeatable. Great. They made amino acids. We do know that amino acids are so important for making proteins. And proteins are the basis of enzymes and what we need for our bodies. However, they sort of glossed over the point that, that little water part of the simulation just turned sort of brown and black. Well, actually it became a gooey, intractable tar, which means it can't move around. It's not very liquid anymore. And that really slows down any kind of reaction that can take place. What they did not see, and this is where it becomes very, very challenging, was there was no evidence that two amino acids came together to form what's called a dipeptide, or more than that, 3 or 4 or 5, to start making a protein. As I constantly hear from my biochemistry faculty colleague, Dave Novak, who teaches biochemistry, and he loves to show me in the biochemistry textbook, when you have amino acids, they don't come together in water. They fall apart in water. So if you have a protein, they'll tend to fall apart in water. So if these proteins or these amino acids are forming in water, or they're being created in water, how do they come to make a protein? Because water actually takes them apart. So they're in the wrong thing. Well, scientists are speculating, well, maybe it was on the edges, so where it dries out and the water is lapping over there. Okay. It needs to be something at least like that, because they will not form in water. This is one of the great mysteries, actually, is how is our body making these proteins in 50 to 70% water? Well, you can design a system when you have the right tools to knit these things together. And we'll see a little bit of that later on in a nice little video. But naturally, they want to fall apart. Just like we were talking about earlier, water and carbon dioxide naturally, when you put them together in water, they make carbonic acid. They will not make sugars and oxygen. But if you have a whole design system, you can bring these parts together and do that. One of the other major problems with this experiment and some of the other ones is that we don't really know what gases were on Earth at that point. But I don't know if you can see it, but in there they actually had methane, CH4, ammonia, NH3, hydrogen, H2, and water, H2O. They're missing a very important ingredient of life in there. I don't know if anybody can tell which actually they're missing 2 big ones. But one important one was oxygen. As soon as they introduced oxygen in this experiment, no amino acids. So there's not very good evidence, geologically speaking, where there was that much time where it was oxygen-free on this planet. And that's a whole other story to get into. But so oxygen, no amino acids. No oxygen, you can make amino acids. Well, that sounds a lot like nitrogenase and what's happening in the roots of legume plants. So there's that. And this only makes proteins. This particular mixture they did doesn't make nucleobases, the bases, the base pairs, and the components of DNA. So, and we're going to see this catch-22 that's always there. So they made a few in the ingredients of life. They did not make really anything more than a few ingredients, a very small amount. So this explanation from the Miller-Urey experiment kind of got people excited that, man, we can make some of these things. But it turns out these aren't that complicated of molecules to make. And so that wasn't and so it was exciting for a lot of scientists. But it's really just getting back to that chemical design idea. If I were to say, all right, got you some sugar, go make some cookies, what would you say to that? Yeah, where's the rest of it? Well, the sugar, if you shake it enough, it will be, and with some heat and some light, eventually the sugar can turn into flour. Well, maybe. But will it turn into chocolate chips? Will it turn into some of the other ingredients? Well, maybe sugar. Well, I think we need some cocoa in there. Will sugar turn into cocoa? No, that won't happen. Will it turn into eggs if you shake it long enough? And this is the problem, is they're not thinking entirely a lot like how chemists should be thinking in putting these together. So, yeah, this is a nice experiment, but it falls very short. So but the idea was, well, maybe proteins got started. But as scientists were thinking about this, they actually quickly rejected this idea that proteins came together first because proteins are programmed by DNA. So they said, that's not going to work. We need to have some way for DNA to get built. And then they said, well, we actually need the proteins to build the DNA. So a DNA first kind of chemical origins model has been rejected as well. And they're currently working on what's called the RNA world, which is this messenger DNA-like molecule that helps to build proteins, and it's a little like DNA, and sort of the first versions of that were proven ineffective in creating living systems or anything remotely close, and so they're on different versions of RNA, and honestly, they're all running into dead ends, and it just gets more and more complicated. And the scientific explanations of what came first chemically, just dead end after dead end after dead end. And we know this because when, Darwin and a lot of his contemporaries, chemistry was very new during that time. They didn't know of all the chemicals, and the study of biology and what's inside of cells was very new. You see, and now that we know what cells and the chemistry and the things that are made, we see these systems there, and I feel like they're asking me and other scientists to believe, look, if we have some of these parts, and you look here in the water part of my little image here with the fish swimming around, you guys see some parts to something in there? What does it look like? A car, right? And so I feel like they're asking me, out of a warm little pond, a car drove out of there. Okay, sure. What if that really did happen? So if that's what I feel like we're being asked to believe, and I'm going to give you some more evidence to back that up. But I feel like it's not even that. You're even asking that not only did a car or some complicated system come together, that system also had to be a system that can reproduce itself. So I feel like you're asking me that a car drove out and it was able to reproduce and produce other little cars. So self-replicating cars, we kind of figured out that a little bit, but with robots making cars and stuff, but I'm glad that they don't. I mean, it's hard enough to take care of the cars that we have, and little baby cars running around, and so that just seems weird. But that would be awesome, and I feel like that's what we're being asked to believe in, that yes, not only did life start, but the whole replication started at the same time. So I don't know if you ever thought about this. So the simplest organisms are similar to they're bacteria. And how many ingredients, how many chemicals are in there? And Carter can put his hand down because he's heard this many times. This is a fun number to memorize. How many chemicals, how many different ingredients are in a cell? The simplest cell. Now I'm not talking about complicated human cells. I'm talking the simplest organisms that are out there that had to come together, or even if it was a small fraction of this number, I'll give you that. But how many do you think? Throw out some numbers. We got 8. 220. We're at least getting closer there. 1,000. We're getting closer. So I looked this up. How many chemicals has anybody analyzed? And I found in a textbook the components of a cell. Molecular Biology, 4th edition, and 2002. And water makes up most of the cell. That's one type of molecule. Well, there's a bunch of different ions in there. We'll count them as 20. Sugars and related molecules, 250. Amino acids, 100. Nucleotides, around 100. Fatty acids, around 50. Other smaller molecules that we're not sure of, 300. And all the macromolecules, nucleic acids, proteins, enzymes, around 3,000. Total maybe around 4,000. 4,000 ingredients. Now, maybe that's an overestimate, and there's scientists who are working on what's the minimum number that we need, and it's probably a little bit lower than that, but even if the number was 50, how do 50 chemicals come together to make a self-reproducing system? And when you don't know that it's a very complicated system, you could say, if it's 3 or 4 chemicals come together, I'll give you that. They could probably come together. But 4,000? No way. I can't even get 9 ingredients to come together on a regular basis in my kitchen to make cookies. I wish, it's probably my own fault, but we do have, it happens enough. So when we start thinking of it from a chemistry perspective and how we have to design them and put together, this is a much harder challenge to happen. So if we look at the timeline of science a little bit, Darwin's theory was published around 1860, and he just said, yeah, there's some phosphoric salts, maybe carbonic acid, ammonia. The periodic table, in its earliest, simplest form, was published in 1869, 10 years afterwards. So chemistry wasn't even that well organized even at that time. So I feel like the whole system has been, let's just turn a blind eye to how chemistry works, and let's build a theory on, just on what we see here. And so this is the problem, because now, as we study even the cell, large systems here, they don't go, oh yeah, it looks like protoplasm in there with a few chemicals. All the biology textbooks are saying, well, when you look at the factory inside of the cell, really, a factory? It's that complicated? Oh yeah. And it's a very efficient, well-operating factory inside of a cell that's taking place. And so that is the chemistry that all had to come together to make that. And so this is a much harder challenge than I think we're being led to believe. That somehow chemicals can come together and just start making things that are close to life, and eventually if you bounce them with enough heat that they'll make life. Now in my class in general chemistry, we start to look at 4 categories of chemical reactions, and if this goes over your head, that's okay. But I just want to illustrate a point here. The 4 categories, they have to do with these 2 terms called enthalpy and entropy, and I've kind of made 2 little images for them. One's a thermometer, and that has to do with what's called exothermic reactions, reactions that come together to make heat. Those are actually very easy, very efficient reactions. And we know from our study of chemical reactions, reactions that tend to want to disperse, we call this has to do with the term called entropy. And if they want to spread out, those reactions are also very likely. So a reaction that's exothermic, creates heat, and spreads out, that's a type of chemical reaction that occurs at all temperatures. And that's often just like things that when you mix them together, water with something, an acid and a base, they react and they produce heat, and the chemicals that are made, they just spread out. That's a very efficient reaction, and it goes fast. That type of thing is, it just works at every temperature. It doesn't matter what's going on around it, it will happen. It will take place. It's what's called spontaneous. Another very efficient and well-behaving reaction is one where the system is spreading out, but maybe it's endothermic. Energy needs to go into it. So this is the second category, and you can relate that to an ice cube melting. This is that type of reaction. An ice cube, if the temperature's hot enough, it will spontaneously turn into liquid water. So it's a phase change, going from a solid to a liquid. So that is one of those categories. Yep, that takes place. That's spontaneous at the right temperature. It has to be at the right temperature. If it's cold enough, ice cubes won't melt. Another category is the reverse of that process, freezing. If you took liquid water and it was cold enough temperatures, it would freeze. And in that system, the molecules are coming together. And that's a big challenge. You're fighting entropy at that time, but enthalpy makes up for it. Energy is given off. That's the little red guy. So I'm into the third category. The last category is when you're trying to bring a bunch of chemicals together, and the system needs energy, it's endothermic, for it to happen. Now these reactions it's not that they don't happen, it's that you usually have to design a way to overcome the challenges. For instance, aluminum. Oh, we love aluminum. It's everywhere, and it's a pretty cheap metal. Well, aluminum oxides are sitting all around the world. In order to make aluminum, there's a lot of reactions where you're bringing the aluminum atoms together and you're getting rid of oxygen and some other things, and you can bring them together. Well, if you design a system, we can make lots of aluminum, and we can make it cheaply. So you usually need, not always, but you typically need a designer for this last category reaction. So you maybe know where I'm going with this. I wonder which one of these 4 reactions do the life-giving molecules fall under? Are they the type of reaction that they just always come together under any temperature? You know, I kind of get the sense that if we don't know anything about chemistry, we would pick that. You say, yeah, I mean, these things are always coming together all the time. DNA, proteins, that's got to be under that category. And the answer is no, they don't fall under that. How about B? If you get the temperature just right, like photosynthesis, maybe if you get enough energy and light, they will happen. No, they don't fall under that category either. What about the part C? At least those go, but if you just keep it cool enough, whatever cool enough means. No, they don't fall under that category either. You see, all the life, I would say the biomolecules of life, like DNA, proteins, RNA, photosynthesis, they all fall under this last category, where you need energy, you're bringing all these parts together, and they do not happen spontaneously. You usually need a designer to bring them together. And I feel like there's other arguments where people take parts of these arguments and say, well, it's entropy, how do you bring things together? Well, if you have enough enthalpy, and there's ways to overcome entropy issues, but this last category, it just makes it hard to make these molecules because entropy and enthalpy are not favored in these systems when they come together. So you have to fight that. You have to think about that. Designers know how to do that. So not only do you have the chemistry and enthalpy and entropy sort of fighting the chemical reactions that are behind all of this, we think about what's needed because inside the cell we know that DNA is the code which makes, and there's this complicated system which I'll show you in a nice video in just a second, where using messenger RNA and tRNA, these things can build and put the amino acids together even in an aqueous or water system. And then eventually, if you do the code right, you can make all these proteins. Well, it turns out all these proteins are needed to run this whole system. And they're the tools that are made to make more DNA and to make more of these molecules. So where did it get started? Do you need the DNA, the proteins, but you need the proteins to make the DNA. So there's this whole system that's in there, and where do you get started? It's worse than the chicken and egg problem. Does the chicken come first or the egg? And people have their own theories on this, but you have all of these pieces that need to come together, and you have this system. Thousands of molecules need to regulate this whole system that's in place there. So I thought instead of talking about it a little bit, let's see this factory at work. And this is a video I found from Stephen Meyer, an intelligent design advocate, and he helped put some of this together, I think, and it's amazing to watch how DNA works and how it's put together. So let's roll the video number 3 here.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
So that's amazing. That is, and I am so thankful for other scientists who have figured this out. It's taken decades and decades, and people with a great imagination and computer skills figuring out how to animate what's taking place there. And they're just showing just some of the functions that's there. So this was taken from Stephen Meyer, and he has a book that I'm going to refer to in just a moment. So how do you get out of this hole of chemical origins? They are stuck down in this hole. How do you get started? Well, they're going to keep throwing money at this problem, and more chemists are going to get thrown at this problem. And we know a solution out of this, and we call them a designer. Because when we're bringing chemicals together in a complex way to create all of these structures that are needed, we see this happening all the time. And so when we say it's so hard and it's so complex that nobody could possibly do it, it needs God, that's actually... we're saying those things, but we want to understand that it's not, we understand that they're complicated, but we know in a positive way how these things get done, and that is through these complicated chemical systems are often put together, and we do that in our kitchen, we do it in the lab, and we call them bakers, we call them chemists. And so when you put minds and intelligence behind them, we can put these complicated things together. So it's actually it's a scientific argument, I believe, when you say, okay, we see people doing this, we see intelligence doing it, and we don't see nature doing it, but when we look at a system, we see all these complicated chemicals come together. We don't have to say, well, it's so hard, God did it. No, we know that intelligence can do this sort of thing. So I believe it becomes a positive argument, not an argument from, well, we don't know, so God had to do it. And I don't think we should be necessarily taking that argument other than to say, hey, we should understand how it works so we can appreciate what God has done. So the third and final argument is one that was alluded to in that video by Stephen Meyer, and it has to do with this idea of information. And I know some of you are familiar with this. And so in just a moment, Carter, I'll need you, because we're going to have a demonstration here. I have some magnetic letters being shown on the screen here. And how do letters come together to create important information? And that's what we want to talk about in just a second. First of all, when I was reading his book called Signature in the Cell by Stephen Meyer, I'll show you that in just a little bit, for some reason awoke in me this new picture of what was happening in DNA. When we look at DNA, as I have it up here on the screen, I want you to focus in on a few things. There is a code in there that we use letters to represent. A for, there's A, T, C, G, and these are the bases that are in there that are connected in the middle of the strand, the base pairs. And they are held together by what's called a ribophosphate backbone. And I thought, oh man, this is very complicated. It's amazing how Watson and Crick and actually other scientists figured out how this double helix structure works. Well, I started to focus on the backbone for a second. I'm like, oh, wait a second. That's the same thing all the way up and down. There's a phosphate structure and a sugar called ribose. And so if I were, and I said, well, wait a second. What if I were to cover up the DNA sequence? Could I figure out the sequence of letters that are there from the backbone, the ribose and the phosphate? And I said, wait a second. I can't. There's nothing about the backbone there that is driving the chemistry to know whether there's going to be a thiamine there or a guanine or an adenine, cytosine. I can't tell. So it's like it's the rungs of a ladder. You can have the piece of wood there. They don't always tell you what is going to be there. So from what I can tell, and I think what most scientists would believe, that there is nothing about the chemical backbone that's driving which bases should be there. Now, we do know a C and a G are supposed to go together, and they do, and a T and an A go together, but you cannot tell who is going to be there in what order. So not only is there that, but just because you have an A and a T together, you can't just guarantee what's going to be next beside these things. And the analogy has been made, and we've already done it before. Carter, you can come up here, that this is a lot like, and I'd like you to hold this up, the DNA backbone is a lot like a magnetic board, and the base pairs are letters. Does the magnetic board here determine the sequence of letters that I see here? No, I can, right, move them around. Can you tell what word is here? Let's see if you can, let's move them around a little bit and then show them. Let's see if they can figure out what word this is. Do you guys know what word this is? What do you think? Here, we'll turn that up. So, okay, hold it up nice. And you guys think you know what word that is? It's hard to tell. This is a word. I just scrambled the letters. But the magnetic board is not telling me which order to go in. My mind is telling what order to go in. And this is exactly what we see inside the cell with DNA. The chemistry is flexible to allow any order of A, T, C, G anywhere. Just like I can put these in any order that I want. Anybody figure out the word yet? Let's give them a hint. Put the first letter in the right spot. And so when we have intelligence can go and dictate the code. Anybody? Oh, yeah, we heard it. Okay, put it, and then we'll say it up that loud. All right, if you understand the code and you understand the information, you can understand what's there. Okay, go ahead and show them. Okay. Gecko, so a nice little chameleon, right? And do the geckos change their colors? No. Oh, but they can do amazing things. What can they do?
Climb walls.
They can climb walls. That's fun to study. Okay, I was thinking chameleon, but gecko, that fits in there. It's smaller. I'm not a good biologist. All right, when it comes to animals. No, I'm learning. Thank you, Carter. We'll have you back up in just a second. So the analogy between the DNA and the magnetic letters, I think, is very appropriate. There's nothing about the chemistry that's naturally there that dictates which order it should go there. So then who decided the order? What decided the order? How did the right order get there? Did it just by somehow getting made? So what if I gave you, number one, fine-tuning the universe, and two, I gave you all the right ingredients that you need to make DNA, to make proteins. Where does the information come to make all the right ones? How does that get there? Even if we gave everything that was all the right ingredients, all the right temperature, how do you get to the right code so that it can do something useful? There are so many ways to arrange these letters. These are just a few letters here, and there's thousands and thousands of base pairs in DNA, and it's easy to, there's more ways to scramble these up than there is to make an actual word that makes sense. This is the exact same thing that we see going on in DNA and RNA, the codes that are there. It's just like what we see in books. It's like what we see in computer codes. We know where books and codes come from. They come from minds putting together information and conveying it in a way that a receiver can understand it. And so once again, we see, I think, another positive affirmation for how intelligence can be identified. When we see codes and we see ways of interpreting that code, that is telling us there is intelligence and an intelligent designer putting together that code and that information in the right order to have a purpose and for life. So let's think about this for a second. We talked a little bit about DNA yesterday and how it gets damaged. I want to bring that back up again just for a moment here. And so inside one cell of your body, if you were to unravel the DNA in there, it's a very, very thin string. It will stretch out 6 feet, which is with the length of my arms here. Okay? Now, how could that be? Well, if you were to take Play-Doh, right, and you start rolling Play-Doh, what happens to it when you start rolling a little ball of it? It gets longer, right? Imagine if you could just keep rolling it down so it got so thin and it didn't break apart. How far could you get? Well, that's how I like to imagine DNA. So it's sort of wound up in a ball, but as you spread it out at 6 feet in one cell. Do you know how many cells you have in your body that have the DNA? They have DNA in it? Well, it turns out there is about 37 trillion cells in your body. If you multiply that by 6 feet, that is equal to 42 billion miles of DNA in one person. And that is the distance that goes from the sun to Pluto back and forth, what's that, about 12 times. That's in your body right now. And if any of that DNA starts to make a mistake, that leads to cancer, and that leads to death. And we know that that is a problem that our bodies can hardly even keep up with now. And this is why cancer is so prevalent, is because we have the repair systems that are in there that are trying to keep up, but they don't, with the radiation and now how we eat, and I think the genetic degradation of our lives, of living systems, and without God's support in a lot of these things, we try to eat well, and drink water, and we do all these things. But when you have 42 billion miles of DNA, and any one of it can go wrong and lead to cancer, it's amazing that we're all not having cancer right away as soon as we're born. And so we need to have a system in place, and that's what we see, that is correcting for any of the mistakes in this 42 billion miles of DNA that occurs in our bodies. And I think that's amazing. Now, I showed you a little bit yesterday on a video. It was a cartoon from a TED Talk, and on how the DNA is repaired. And I just think that's amazing. I thought maybe we might show one more video on that in just a second. Once again, if those weren't here, Nobel Prize in Chemistry for the scientists who have worked for 30 years to figure out how there is at least some way to repair some of that. And unfortunately, for many people who have suffered from cancer, and it happens from the youngest person to the oldest person, it feels like the longer we live, it just gives us a greater opportunity to get cancer. And I feel like with all the money that's being spent, billions and billions of dollars, we are hardly making a dent in solving this problem of cancer. And cancer is not one thing. Any one of your cells, of your 200 different types of cells, can go wrong in a variety of chemical ways. And so it's like hitting a moving target. And science is making some progress, but it's too little, almost too late, unfortunately, for many people. So this problem with cancer, it would be way, way worse if God did not have some process in there stopping some of it. And I wanted to just watch it one more time in a little detail. This is not to say that cancer is an easy thing. It's actually what is happening inside of our cells. I just want to continue to impress upon you how complicated the systems are in there and how many pieces have to come together for life to work and to keep the problems from amplifying. So we're going to watch one last video. This video is actually, someone's tried to simulate just one of the repair mechanisms that's happening in bacteria. Because if you didn't even have bacteria, we would wipe out bacteria in just a little bit. So let's roll the last video.
This is a video of a 3D animation on the mismatch repair pathway in E. coli. In the cytoplasm is the E. coli's DNA, the molecule that contains all of its genetic information. DNA is made up of nucleotides, each of which contains a phosphate sugar backbone and an organic base. The two strands of DNA are held together by hydrogen bonding between complementary base pairs. Adenine pairs with thymine and cytosine pairs with guanine. DNA mismatches occur during DNA replication. For simplicity, this animation will show DNA as a flat molecule. Before replication can begin, a helicase needs to unwind the DNA molecule. In E. coli, DNA polymerase III catalyzes the incorporation of new complementary bases to the new DNA strand. This is only the alpha and beta units of the DNA polymerase III enzyme. However, it is possible for DNA polymerase III to incorporate a non-complementary base to the new strand. This is known as a mismatch. A mismatch could result in a faulty protein being coded for, so it needs to be repaired. The first step in the mismatch repair pathway in E. coli is recognition of the mismatch by the mut-S homodimer. Mut-S recognizes the mismatch and binds to it. Then, the mut-L homodimer binds to mut-S, ready for a later step in the mismatch repair pathway. Meanwhile, up to a kilobase away, the mut-H protein recognizes a GATC site. Mut-H is a weak endonuclease, which will create a backbone incision between the guanine and adenine residues on the newly synthesized strand when activated. Mut-H is able to differentiate between the newly synthesized strand and the template strand by the lack of methylation markers on the newly synthesized strand. This is why the pathway is also known as methyl-directed mismatch repair. In order for mut-H to become activated, it needs to bind with mut-L. This causes the DNA to bend. The newly formed backbone incision allows an exonuclease to enter and begin excising nucleotides from the newly synthesized strand. The exonuclease excises the mismatched nucleotide and stops excising just beyond it. Now, DNA polymerase III can begin to resynthesize the new strand, inserting the correct complementary base where the mismatch used to be. The final step is for DNA ligase to ligate the backbone nick. The mismatch pathway is now completed, and all the newly synthesized DNA is complementary to the template strand.
Even fixing one of the small breaks in there, how many different pieces have to come together to fix that one thing? And I want to remind you again, not only do we have the problem of information in DNA, we have a system in there that's fixing the information if something goes wrong. So it's amazing enough that there's a system that has a code in there. But there's another set of systems in there that is correcting it to make sure the right words are always in there, in the right order, so that the body knows to make the right proteins and things. And that's amazing. So there's a correction system, and we know the correction system has problems at this point in Earth's history, and it's not always working that great, because we still get cancer, but it's in there, and this has surprised a lot of people here over the last few decades to see this. And so when we look at all these complex systems, we see the information, we think about how can chemicals come together to form life. I think there's so much evidence that's pointing to intelligence in a designer that had to come in to put these pieces together. So here are some of the books that I've been reading or have read. Signature Nacelle by Stephen Meyer, and these gentlemen, Michael Behe, Stephen Meyer, they worked for the Discovery Institute, and they don't necessarily are saying who the designer is, but how we can identify, and it's legitimately scientific, how to think about how designers participate in chemical processes. So once again, the problem with having these three holes is how do you get out of them? And if you don't get out of them, you don't have life. You're dead. And I think these are the three biggest ones: the fine-tuning of the universe, chemical origins of life, and the information that we see there. I think these are great big holes that there is not good chemical and scientific ways to get out of there. Now, scientists are going to keep looking for it, and we'll keep seeing articles published in these things that are produced. And I want to do one last experiment with my kids. Come on, if you would come up here. And I have a Bible verse talking about if you see a house in Hebrews here. I'll just read Hebrews 3 and 4. For this one has been counted worthy of more glory than Moses, inasmuch as he who built the house has more honor than the house. For every house is built by someone, but he who built all things is God. You can't have a house unless you have someone building the house. And the house that we're living in right now, Earth, I'll have you go right here, and then we'll put you here, and you're here. Now, this reminds me of what we're hearing about scientific information. Reminds me a lot what took place when Moses went in front of Pharaoh. And, of course, he said, "Let my people go." And Pharaoh said, "No." And the first plague came upon the Egyptians, and the water turned to blood. And I can imagine there that it says that Pharaoh asked his magicians to come and see if they could explain what was going on. But I think those magicians were probably a lot like chemists. We probably would call them scientists today. They were called, because a lot of what we do seems like magic. In fact, we're going to do one last magic/chemistry thing for you here. And I can imagine Pharaoh going, "All right, you guys, you're my wise men, you're my magicians. Have you figured out how water can be turned into blood?" And they said, "Yes, they know." And they said, "We have figured it out. We have some special ingredients here. We can take water, and we are going to show you, O great Pharaoh, how to make blood from water." And so I can imagine they had their potions, they had their chemicals together, and then let's go, yeah. So they poured into the flask there. "Oh great Pharaoh, this is how you do it." And so they pour it in there, and there you go, pour it right together. That's good. It might get too dark in there. And they said, "Oh, great Pharaoh, we have made blood." And this is actually an iron compound, by the way. And then I'm going to put it back down on the table. And so that is taking water and turning it into blood. And Pharaoh and the others said, "Oh, if we can do it and show it scientifically, then we don't need God anymore. So we proved it. We don't need you. Thank you, all great magicians and scientists. You can go back and sit down." Appreciate them. And I feel like we've got to that point in Earth's history where we're saying, "Oh, we can explain it. We don't need God anymore. We know how DNA works. We don't need God anymore. We know how a cell works. We don't need God anymore. We know how to make chemicals come together. We don't need God anymore." Really? I mean, we still needed a lot of things to come together. And when we see a system, we see a house, we know that house must be built by someone, and he who built all things is God. I want a renewed look at these things, not to go, "Well, it's so complex, we don't need to study it, God made it." Let's get in there and see how God made it. And we will see his handiwork all over it. And I love to be able to study it because we get to learn about God, our Creator, when we do that. And I'm going to quickly read here in the last couple minutes a quote from Ellen White here in Patriarchs and Prophets, page 45. And I couldn't agree with her more. This is in 1890. She wrote, "Here is no mystery. There is no ground for the supposition that man was evolved by slow degrees of development from the lower forms of animals or vegetable life. Such teaching lowers the great work of the Creator to the level of man's narrow earthly conceptions. We're so intent upon excluding God from the sovereignty of the universe that they degrade man and defraud him of the dignity of his origin. He who set the starry worlds on high and tinted with the delicate skill the flowers of the field, and who filled the earth and the heavens with the wonders of his power, when he came to crown his glorious work, to place one in the midst, to stand as the ruler of the fair earth, did not fail to create a being worthy of the hand that gave him life. The genealogy of our race, as given by inspiration, traces back to its origins not to a line of developing germs, mollusks, or quadrupeds, but to the great Creator." I am so thankful for those words, and this is exactly what we're seeing, I think, in science. And so what I'm here to testify today is science and scientific information can be used in a lot of ways, and unfortunately it's being used in a way to lead us away from God. But I see, as we dig deeper, God's handiwork all over this. And the more that we share this information with each other and teachers with students, there is plenty, if you love science, there's plenty of scientific information to underscore that there is a designer there, someone who put this together. We do not need to resort to, well, time and chance is enough. No way. The chemistry is pointing away from that. So we are without excuse, folks. And I think the whole world is, and we read that in Romans 1:20. For since the creation of this world, his invisible attributes. See, this is why we need chemists, because chemists help us to see the invisible attributes of the world. We can clearly see them, being understood by the things that are made, even his eternal power and Godhead, so that all are without excuse. Because although they knew God, they did not glorify him as God, nor were they thankful and became futile in their thoughts, and their foolish hearts were darkened. You know, if you had a friend and they made this great car or the great thing, and you walk out there and you say, oh, that's cool. How did all those pieces just kind of come together? No, you would go, hey, that was really neat. You probably put a lot of work into that. I am so thankful on how you designed it and put that together. But when we look at this world, we go, I don't know how it works, so it must have just all come together by chance, and we have the stars tell us we've got enough time, and we see animals changing, and so it all must have just happened by chance. That's not what we see when we dive deep and we look at the invisible things that are there, God's invisible attributes. They're all over this place, and they are pointing right back to a designer, and I think that's what I see as a scientist. So I thank you for going with me in this journey. And I think there is a ton of scientific evidence that points to a creator. And that's just the tip of the iceberg, I can assure you. I was trying to make it simple enough, and the deeper I dig, the more I see there. So thanks for hanging in there with me, and I hope we can turn the scientific information back to show that God did do it, and just because we have a scientific explanation for it doesn't mean we can kick God out of the room. But we now have a way to appreciate how he did it. Let's end our time with prayer. Father God, all I can say is thank you. What an amazing world that you have made. And we get to see all the wonderful ways that you have done it. And there are so many ways that we can attribute your handiwork. And I pray that others will come to have faith in you because they see that and they trust it. Well, if you trust science, well, then science is pointing us back to you. And so, out of all of that, Lord, your word has been sustained through nature and through Scripture, and they all point back to you. So I thank you. May we not be ashamed of what we believe. We are not abusing our kids. We are giving them good scientific information and good ways to think that point back to you, our Creator and our Designer. And we should not be afraid of this. So thank you, Lord. Bless our time. And others, as we leave here, may we be encouraged and strengthened to know that you are there. We can trust you with every aspect of our life. So please go with us, Lord, and may we be bold witnesses for you. I ask this in Jesus' name. Amen. Thank you, guys. God bless you as you go to your other meetings.